Welcome to the third contributed talk session. We have 4 wonderful speakers today. Each will be giving a 10-minute talk, followed by a roughly 15-minute Q&A session. Please submit your questions via the Pathable poll and vote on other questions that you would like to have answered. Also, please remember to note which speaker the question is being addressed to. That'll help us ask it of the right person. And so the first talk today is by Lukas Weber.
Thanks very much for having me here to talk today. So I'm a postdoc at Johns Hopkins University now. I'm going to talk about work we've been doing on developing unsupervised analysis pipelines for spatial transcriptomics data in human brain. And we've just had, I think, the keynote talk before this about spatial transcriptomics, since it's great timing. Some background here. So in spatial transcriptomics, we're measuring transcriptome-wide gene expression with the spatial context recorded of each measurement. These illustrations here are from the 10x Genomics website showing mouse brain. So on the left, this is a histology image of half of a mouse brain. Then the way spatial transcriptomics works is we've got a grid of so-called spots located across the tissue slide and measuring gene expression at each of those spots. In the middle, this is showing the total gene count at each spot in this experiment. And you can already see that that's matching to some of the morphological structure in the mouse brain. And then downstream of this, we can do all sorts of analysis like clustering on highly variable genes or looking at specific cell types, doing an analysis for certain cell types. And this has really been enabled recently by several new technological platforms that have been released. We've been working with data from the 10x Genomics Visium platform, which was commercially released late last year. Here are some more schematics showing how that works. So again, on the bottom left, this is a mouse brain tissue slide, then showing how this grid of spots is overlaid on top of the slide. On the right, that's showing the workflow. So the slide has 4 capture areas, each about 6.5 by 6.5 millimeters. Each of those capture areas has around 5,000 spatially barcoded spots. And then you're getting reads that are both spatially barcoded and with UMI barcodes as well. And then after this, the analysis has quite a lot of similarities to single-cell RNA sequencing data. So as a first approximation in our project, we sort of assumed that 1 spot is equivalent to 1 cell. Now that's quite a rough approximation. In our data, it was more like 3 cells per spot on average, and quite a distribution from 0 up to about 10 or so. But using this approximation, we could then reuse all sorts of bioconductor infrastructure and pipelines for single-cell RNA-seq analysis, which was really great. So on the bottom left, that's a schematic of the SingleCellExperiment object structure. And I think in one of the next talks, Dario Righelli is going to talk about their extensions to that for spatial data. And on the bottom right, this is this orchestrating single-cell analysis online textbook, which was initiated by Stephanie Hicks and maintained in large part by Aaron Lunn. And we can reuse many of the methods from there. We got involved with this work through an experimental collaboration, a really nice collaboration together with people at the Liebe Institute for Brain Development. I won't go into too much detail here. I've got some links on the next slide to a poster by Leonardo Collado Torres and also a preprint that we've put out. So this work was interested in defining the spatial landscape of gene expression in the human brain using the new Visium platform. Some key points there on the left is that the cortex has 6 known layers plus white matter, and we can do analysis like testing for layer-enriched expression signatures between those layers. Also linking to published data, spatially registering existing single nucleus data, integrating with neuropsychiatric gene sets, and then doing both supervised and unsupervised analysis pipelines. And my work was largely on the unsupervised pipelines. On the right, that's the experimental design from this project. There were 3 neurotypical adult donors, 12 samples in total. On the top right, that's showing what the actual slide looks like, and with those 6 layers and the white matter below. At the bottom 3 panels there are showing genes enriched in the white matter versus the neurons versus specific layers, which is what we're looking for. And yeah, links here. So Leonardo Torres just presented a poster in the previous poster session. We've also got a preprint out and also really nice online resources for exploring the data and the results there, also maintained by Leonardo. And on the bottom left are our collaborators from the Libre Institute. Now, in the unsupervised analysis pipeline, so this was what I worked on together with my advisor, Stephanie Hicks. So why doing unsupervised analysis? And the reason is, so in this project, we had our collaborators doing manual annotation of those spots to the known layers. This is very labor-intensive work, and in other tissue contexts might not be possible at all if there's not a known morphological structure, for example. So having an unsupervised analysis method lets us do exploratory analysis as well and look for unexpected results. This is showing the overall sequence of that pipeline. So first, starting with the tissue sections, looking for spatially variable genes, clustering on those genes, and in this data set, we can compare against the known ground truth from the manual annotation. And we built this up by adapting bioconductor-based pipelines for single-cell RNA sequencing from the OSCAR workflow, adapting that to the spatial data. So going through standard steps such as pre-processing, normalization, feature selection, dimension reduction, clustering, and here having those known ground truth labels. We adapted especially in the feature selection part. So there are new methods available for testing for spatially variable genes instead of just variable genes. And also within the clustering, we added spatial coordinates as additional features for clustering to see if that could improve performance. And this is showing some of the results. We did several styles of data-driven analyses, including the completely unsupervised, which is on the left, doing clustering on the spatially variable genes. We also did a semi-supervised analysis workflow where we used genes from the manual annotation. And separately, we also used known marker genes from the literature as a baseline comparison, and comparing this against the ground truth. So this is showing those 6 layers of the white matter below. Then we tried lots of different clustering implementations to see if we could optimize this. So there were questions of which set of genes to use, whether we use that spatial DE method or highly variable genes. Question of which type of dimension reduction to use, principal component analysis or UMAP. A question of whether to include the spatial coordinates as additional features or not. And in each case, we compared this against the known ground truth from the manual annotation. We found in the end that including the spatial coordinates slightly improved the clustering performance. So here we're showing the adjusted rand index from the known clusters. Each point is one of the 12 samples in the data set. Each set of colors is showing with and without the spatial coordinates included or not. Unsupervised work on the left. Using the spatial DE genes did not improve performance very much compared to the highly variable genes. We're working on that. And finally, it also let us find unexpected results, which was very cool. So on the right here, we've got 6 genes. The ones at the top are known laminar genes from the layers. The ones on the bottom are ones that we found in the unsupervised pipeline, which were spatially variable, but not linked to the layers. And these are in fact genes that are linked to things like blood vessels, and immune cells, which we were not really expecting to find in this data. On the left, that's showing the test statistics between comparing the spatial DE test statistic against the manually annotated ones, showing that for these unexpected ones in red, we get a high test statistic from spatial DE, but low from our manual annotation. Okay, and the last thing I just wanted to quickly mention is we're now using this as the base for developing a longer and more comprehensive workflow for spatial transcriptomics unsupervised analyses using Bioconductor, and this is very much a work in progress, but can keep an eye out for this. This will be released in this similar online textbook format as the existing OSCAR book. And that's it. So thank you very much to my postdoc advisor, Stephanie Hicks, and to our collaborators at the Libre Institute on the bottom left. As I mentioned, there's a poster which is now available online from Leonardo Torres, and also our preprint available online. And that's it. Thanks.
Wonderful. Thank you for a very interesting talk starting us off. For some of the attendees that maybe came in late and missed my opening spiel, please submit your questions using the Pathable poll and make sure to denote which speaker you're asking your question of. The next talk is by Dario Righelli, and it's a pre-recorded one.
Hello everyone, I'm Dario Righelli and I'm a postdoc in the resource lab at the Department of Statistical Sciences at University of Padova. And today, as you can see, I changed a little bit my presentation talk because I extended the Visium Experiment class with other classes. So we decided to create a unique package called Spatial Experiment, which gives multiple S4 classes for spatial transcriptomics. With spatial transcriptomics, there are several technologies which provides spatial coordinates of the cells referred to a reference, in our case a tissue or a portion of a tissue, and moreover they provide also transcript quantifications. Of course, the number of transcripts varies depending on the used technology. We focused just on two technologies at the moment, the SecFish data and the 10x Genomics Visium Transcriptomics, but there are several others, and some others they are coming out. The SecFish combines microscopy imaging with multiple rounds of hybridization combined with temporal barcodes in order to catch the coordinates of the cells on the tissue. And while the transcript abundance is detected as the number of colocalizing spots per each transcript. These can be combined with single-cell RNA-seq datasets to have single-cell multimodal integration. The 10x Genomics Visium transcript is a little bit different because the tissue is placed on a chip with several spots on it, and while we catch the image of the tissue on the chip, some cDNA amplification is made inside of each spot. So in the end, we will be able to detect the spot thanks to its barcode and to have the transcript abundance associated to that specific spot. We started from the single-cell experiment class because it sets a standard for any kind of single-cell data storage and analysis in R. It is in Bioconductor since the version 3.6, and it provides several attributes and methods for the handling of this kind of data. So our package provides two S4 classes, the Spatial Experiment class and the Visium Experiment class. The first one is more general for any kind of spatial experiment data, while the second one is specifically designed for the Visium data. At the moment, it is available on just on my GitHub, but we are under Bioconductor revisions. And if you are interested, the sticker is available also on my GitHub at the moment. The class, the Spatial Experiment class, inherits from the SingleCellExperiment class, as mentioned before, and it wants to set the basis for the spatial transcriptomics, for the spatial experiments analysis, and we implemented some attributes named spatial coords. Typically, they are X and Y. They are stored inside this part of the single-cell experiment class. And additionally, of course, we designed multiple methods for getting and setting these attributes. We are still working on other methods for this class, such as the cbind method, where we will be able to bind to spatial experiment objects. In order to give an example of usage, we used a dataset of seq-fish and single-cell RNA-seq data provided during an hackathon for the BIRS biointegration workshop, and the usage is available in this other repository named Spatial Analysis, and you can find the seq-fish spatial analysis HTML workflow here in order to understand how to load the data from file and how to store them inside the single-cell experiment for the single-cell RNA-seq and into the spatial experiment for the seq-fish data, and how to manage to play with the class here. After that, we extended this class with the Visium Experiment class because the Visium Transcriptomics provides multiple informations, such as for the spatial coordinates, it provides X, Y, in tissue, the full resolution of the pixel, and for columns and for rows. And additionally, it provides multiple images in different format, and to interchange between these images, you need some scale factors. So that's why we provided also the methods for handling with these other attributes, and we provided also a class usage always in the same repository, but with a different name. Here in this case is Visium Analysis, and we used a dataset provided by the 10x. It is available on the 10x site, genomics site, and here there is a tiny URL because it was very long to put it here. Also in this case, we show how to load the data. There are very it's a very high number of files, so in different formats. So we show how to load this data and how to store them into the Visium Experiment objects, and how to get and set spatial coordinates and image paths. Finally, we used our Spatial Experiment class into the SingleCell Multimodal Experiment Hub Bioconductor package. This other package has been implemented by Marcelo Ramos in the Valdron's lab, and it wants to the main idea of this package is to store and give access to multiple data sets of single-cell multimodal type. At the moment, there is a single-cell NMT-seq data set, and I provided to contribute to this package. I contributed to this package by implementing the function for storing the seq-fish plus single-cell RNA-seq dataset. The main idea is to have one single function for each kind of data. For the seq-fish, we have this seq-fish function, You can select the data set of your interest by aid of the data type argument. In this case, the name of this data set is mouse visual cortex, and each of these functions returns a multi-assay experiment. Indeed, as shown in the figure, the segfish data are stored in the spatial experiment, while the single-cell RNA-seq data into the single-cell experiment, and we combine them into a multi-assay experiment object. In the next feature, we want to allow the interoperability between the spatial experiment classes and some other external works, such as the Giotto tool of Ruben Reeves, the Spatula package of Kevin Roux Albrecht, and some other scripts for the spatial visualization developed by Sheila Gazanfar. Additionally, we want to implement some dedicated attributes and methods for networks, because networks interaction is a very important step during the spatial analysis, and we want to build a dedicated package for the with spatial analysis examples. Moreover, in our lab, we are working on the implementation of some other specific analysis tasks, such as clustering, in order to release them in other dedicated packages. Thank you for your attention, and if you have any questions, I will be here for the question and answers session. Thank you.
Wonderful. Thank you for another great talk. Our third speaker today is Lambda Lu, who will be speaking live.
Hi, my name is Lambda, and I'm a graduate student at Caltech in biology. And so basically, this project started as the effort to write a review paper in spatial transcriptomics. However, it has grown far beyond its original scope. So basically, so here I curated a database of literature in spatial transcriptomics and did some in-depth analysis of the metadata of the literature inside this database. So in this presentation, I will show some highlights from the analysis of the metadata, as well as just a little bit about the data from different spatial transcriptomics technologies from the mouse brain. So some of you here probably already know that there are some databases of resources for single-cell RNA-seq. For instance, Awesome SingleCell and the sdRNA tools for which are databases for single-cell RNA-seq data analysis packages. Also, Valentine, who used to be a postdoc in our lab, curated a database of literature in single-cell RNA-seq and did some analysis on that database to find some trends in the single-cell RNA-seq field. However, so far I'm unaware of any such database for spatial transcriptomics resources, and so this is what this project is about. So first, there's the curated database of the literature in spatial transcriptomics, and it's publicly available on Google Sheets. So far, there are over 400 papers in this database, and includes some historical literature to show some historical context from which this field emerged, and also shows the data encode availability, species, and tissue of data sets, programming languages used to perform the downstream analyses, and to build the data analysis tools. And yeah, there's a sheet in this database dedicated just to data analysis tools. And so with all this data here, I did some analyses. So all the code and the notebooks for the analysis are put into our package called Visium ST, and so far it's not on CRAN yet, and it's only available from our lab's GitHub. And so examples of these analyses are the popularities of programming languages in this field, and where spatially is spatial transcriptomics research performed, and also because the for LCM or laser capture microdissection, they're just too much for curation. I did text mining of the abstract, which includes like topic modeling to find different topics. And all these analyses and code can be explored interactively on RStudio Cloud from this URL. So here we do see the y-axis is the number of publications per time bin, and so here we see the remarkable growth of this field, especially since 2018, and but so for quite long, so if the number of publications indicate interest in the field, then for quite long, interest in collecting data or developing the technology to collect data in spatial transcriptomics was kind of ahead of the interest in data analysis. However, data analysis has been catching up recently. So maybe you wonder what's that? That's voxelation. So in the early 2000s, so some researchers at UCLA cut up just sections of mouse and human brains into like pixels or like grids of cubes, and then did microarray on those. And and then in the first half of the 2010s, we saw some of the more familiar methods already introduced in the previous talks in this session, such as seqfish, merfish, and then ST, which is spatial transcriptomics with capital S and capital T, was introduced in late 2016. Then, but since 2018 boom, we have all these new other new sorts of technologies. So especially recently, given this growth, so which of these technologies are more commonly used? And we see here that the ones that see the most growth are like ST and Visium, which are array-based and not single-cell resolution. But for single-cell resolution ones, we have ISS and merfish were used more recently, and there is seqfish, but however hasn't been used very recently. So also for all these different methods, so how the quality of their data compare. Here I plotted the experiment or data set versus the number of transcripts captured per cell. And for the array-based methods that are not single-cell resolution, this is normalized by the number of cells in each spot. And we see here like slightly captures the not as many transcripts, and the ST and Visium are pretty comparable to single-cell RNA-seq, while seqfish plus introduced in the previous talk captures more. So also how does this compare among the various different single-cell resolution spatial technologies? Highlighted here in red is the seqfish plus here. It's the same one, and we see that for there are many other technologies that are single-cell resolution that do capture even more transcripts in cells than seqfish plus. So well, this data is so good, then like how are people actually using it? Actually now it's possible to use these single-cell spatial technology to profile like about 10,000 genes. However, not everyone goes for that, and most commonly it's only used to profile a select panel of genes. And even though like it's been shown that especially for merfish, it's possible to use these single-cell resolution spatial methods to profile like say here it's like 2 million cells, and then here it's still like like hundreds of thousands of cells. However, like without those few outliers, usually the number of cells isn't really much greater than like a typical single-cell RNA-seq data set. And so why? Because for the single-cell resolution spatial methods, there are many challenges. For instance, you have like terabytes of imaging data, and but there isn't an image processing pipeline designed for these methods they're efficient enough and or universal enough. And that's one reason. Another reason might be that the probes needed to profile so many genes can get quite expensive. And so these are about data. So how about data analysis? So here, so this plot shows the top. So which types of data analysis are people are the most interested in over time. And So here again, the gray histogram in the background shows it's for like all the different types of data analysis tools. Yeah, so again, we see consistent with the observations from like how we're actually using the single cell spatial technology. That the most popular ones are actually designed for integrating the spatial data with single cell RNA-seq data. Yeah, in some ways those are complementary. Yeah, but besides those, we there are a few. There are also like a sort of more and more packages for exploratory data analysis, such as Giotto and ST utility, which is for ST data, and yeah, also like Serata also does that, and then also image preprocessing. Even unfortunately for image preprocessing, the different teams tend to write the pipeline just specific for their own technology, but unlike say STAR, so like for single cell RNA-seq, we have STAR, we have Callisto, which can be applied across different technologies, and yeah, among the other, there are some like more downstream analyses, like spatial variable genes and like spatial domains in the tissue and cell interactions and so on. And so in summary, yeah, so in the highlights of the analysis, yeah, we see that single cell spatial data is often used to complement single cell RNA-seq data, and yeah, yeah, it suggests that the scaling up the single cell spatial data technology can be challenging, and yeah, so also this database can be a portal to discover spatial transcriptomics data sets and analysis tools for those new to the field. And yeah, also there are many more analyses on this metadata available on RStudio Cloud in the URL earlier. And so again, I thank my PI Lior for his support for this sort of sociological and unusual project. And so yeah, and thank you for listening, and I can take questions.
Wonderful, thank you for a very interesting talk. I'm sure there'll be lots of questions. The last talk in this session is by Ellis Patrick.
I hope the other talks in this session have stoked your excitement for spatial omics data. Not just because of the awesome science these technologies will enable, but also because of the exciting analytical and computational challenges that will come along with them. And so in this talk, I'll be giving a big picture overview of two analytical methods we've developed for quantifying spatial relationships between cells. On the right here, we have a standard 3-color image of cells in some colorectal tissue. While we've been able to generate images like this for decades, over the last 5 years, there has been a surge in high-parameter imaging technologies that are able to characterize cells with tens, hundreds, or in some cases thousands of RNA or proteins. The ability to deeply phenotype cells means that we can now pinpoint the locations of a ridiculous number of different cell types, giving us an unprecedented resolution of cell type distribution throughout a tissue. And so what are some of the key questions we can ask with spatial omics data? Well, first, as these technologies don't have a dissociation step, it's arguable that they give us a more reliable estimate of the true composition of cells in a tissue. Secondly, and probably most relevantly, we are able to see which regions of a tissue cell types are localized to and hint at other cell types that they might be interacting with. In this toy figure, we can see that the red and blue cells seem to be chilling out near each other, and the green and purple cells also appear to be co-localized. And so extending on this, if we're able to quantify how cells are localizing in a tissue, we can also start to test if the spatial relationships between cell types are changing under different conditions. Fortunately, there are a range of statistical methods which can enable us to do this. If we have a technology like SeqFish or IMC, where we're able to identify cells, annotate them, and assign them an X and Y coordinate, we can use point process models to summarize the spatial arrangement of our cells. I believe you may have already seen the K function in this session. However, just to revise, the K-cross and L-cross functions are one class of summary statistic that allow us to quantify if two cell types appear to be spatially attracted or repulsed from each other. Put simply, they just give you a scaled average of the number of cell type J within a certain radius of cell type I. On the right, you can see an L-cross function summarizing the spatial association between CD11C positive cells and HIV virions. The diagonal line here are the values of this function if the cells were randomly distributed throughout the tissue. However, the black curve is what we observe in our data, and this indicates that this cell type appears to be localizing in the same regions as HIV. On their own, the K function, or any other measure of spatial localization, only gives us a measure of the association in one tissue, but doesn't tell us how that association may change. Here we have some data from one of the Boda-Miller group's manuscripts. Specifically, we have a box plot of measures of spatial association between gamma cells and T helper cells in both non-diabetic individuals and individuals that have recently developed diabetes. On the y-axis, our measure of spatial association is the area between the L function and its diagonal line. So positive values indicate that cells are localizing towards each other, and negative values means they're avoiding each other. In this data set, we effectively have multiple images per person represented by each point in the box plot. Interpreting this box plot, it appears that gamma cells and T helper cells might avoid each other in non-diabetics, but this relationship seems to disappear in early onset patients. However, there are some complications for formally testing if these changes are random. If we were to plot the measure of spatial association between the cell types against the number of those cell types in each image, we can see that the association metric becomes much more variable as the number of cells decreases. This shouldn't be surprising, as the variability increases as we have less information. And so we propose modeling this relationship and including it as weights in a mixed effects model, effectively propagating the variability from when we generated our summary statistics through to our hypothesis test. And of course, if I design my own simulation for testing for changes in association between two cell types, where I randomly vary the number of cells, we find that including these weights in our test is beneficial. So with these tests, we now have a robust method for testing for changes between all pairwise combinations of cell types. What I'd like you to take away from this heat map of changes in association is that with only 10 cell types, understanding and contextualizing changes in association is not trivial. While we can see that there might be two broad patterns here of attraction and avoidance, if we were to start to classify 20 or 50 cell types instead of the 10, it would probably start to get really tough to begin to interpret these kinds of results. And so because of this, we also propose a method for summarizing broad patterns of spatial association between multiple cell types simultaneously. In our toy example, ideally we'd like to be able to identify regions where red and blue cells localize, and regions of tissue where red, green, and purple cells localize. So briefly describing our method, for every cell we calculate L-curves between that cell and another cell type. This gives us a data matrix where for every cell we have a summary for how close that cell is to other cell types over a range of radiuses. And then we can then take this association matrix, cluster it, and identify cells that have similar localization patterns. We can illustrate this method in an awesome data set from Gary Nolan's group, where they applied codex imaging with 30 parameters to identify 26 cell types within spleen tissue. Here we used our method to cluster all of the cells into 5 localization clusters. The colors in this heat map represent the relative enrichment of each cell type in each of the regions. The first thing we can see is that if we use our method to identify 5 clusters of cells that tend to localize with each other, then we find 5 clusters. The second thing we notice is that there are clearly some regions that appear to be enriched for lots of different cell types and some that are enriched for 1 or 2 different cell types. Regardless, I would like to claim that our method is doing a pretty good job at simplifying the spatial associations between our 26 different cell types. Importantly, if we compare the regional clusters that we identified on the left here to those that were manually annotated by a pathologist in the manuscript on the right, our clusters appear to do a reasonable job of replicating the expert annotation. And so there are some strong advantages to conceptualizing this type of data as a point process. The scaling inherent in the L-cross functions provide the opportunity for all rare and abundant cell types to contribute to the clustering. The theoretical expectation and variances of the L-cross function make it possible to standardize the L-cross function and include summary statistics over a range of radii. And finally, point process models have an established methodology for accounting for tissue inhomogeneity. And so if you want to reduce the impact of large-scale tissue structure on the clustering, this is possible. I'd like to quickly finish by acknowledging my really talented postgrad student, Nick, who has spearheaded a lot of this work, and our bioinformatics group at the University of Sydney. I very much hope that I can clarify any questions in our Q&A session. Cheers.
Wonderful. All right, well, first off, I'd like to thank all 4 of the speakers for 4 very interesting talks. Please keep the questions coming in, and we'll get through as many as we can. The first question is for Lukas. How did you include the spatial coordinates in your unsupervised clustering?
Yes, so in the simplest possible way, which was basically concatenating on 2 more columns of features for the XY coordinates. We, so say starting with the 50 principal components or 10 or however many, then 2 more columns for X and Y features. Now we did test around with scaling the X and Y coordinates to a range sort of comparable to the top PC or the top UMAP. When we used UMAP.
All right, so the next question is for Dario. Can you see Spatial Experiment, the class, also holding morphology information, cell masks, or even images?
Yes, I think that we can handle this because the spatial information are stored into S4 vectors data frame that can load any kind of R class. So I think that we didn't test it, but it could work. But for the images, I don't think that you can, at the moment, we didn't load images in our classes. We just store the path. So it's up to the user to load the data at the moment. But in the future, we want to better handle and try to find better ways to store the images, also big images, I think.
Great. So the next one isn't so much a question, but I think Leo checked out the Museum ST GitHub repo, and Lambda, he's made a note. I don't know if you can see it in the poll, but there's kind of a helpful suggestion there. So maybe I'll ask a question, which is, when you were going through this sort of, I think you called it a sociological analysis, what was the thing that you kind of found the most surprising or was the most different from what you were expecting when you started out?
Okay, yeah, it's a really good question. So, okay, so one thing is, I'm not sure, like, it's not really surprising. I just think like Sweden has played a very big role in this, even though they did not play a very huge role in like in situ atlases before, which is the historical context from which this field arose. Okay, I don't think that's very surprising. Okay, let me think about it. Yeah, I think also, yeah, I think it's also how widely laser capture microdissection has been used. It's also very widely used in plants. Like the plant part wasn't very expected. Yeah, you know, like the more modern like sequencing or SM-fish-based methods are more focused on humans and mice. Yeah, I think that's, yeah, I can't recall anymore. So
No, that's great. So the next question is for Ellis. Can you give more info on the spatial statistics? Were they computed on polygons, and what distance measure was used between shapes?
Okay, for everything I do, I assume that we've used technology, we've got the resolution to be able to segment cells. So if we can segment the cells, I don't use polygons. I just take the center of the XY center of the segmented cell. And so it's a pointwise analysis. And then in terms of distances, these K functions are essentially just taking radiuses around the centers of the cells, and so you can do that over a range of distances. So we're not doing any polygon stuff where we're looking at cells touching, we're just looking at distances between centers.
All right, so going back to Lukas, you mentioned that the histology image matches with the total gene count. For example, in standard single-cell RNA-seq, this could be considered technical variability. Think about Stephanie Hicks' biostats paper looking at detection rate. Is this possible confounding between biological and technical variability problematic in these data sets? If yes, how would you tackle that?
Yes, yeah, that's a great question. And yes, it probably is an issue. And in fact, this is sort of the direction we're going now to try to develop methods to handle things like that. I think especially normalization in this kind of data is a completely open question at this point.
Dario, how would you deal with different quality versions of the histology images? In our case, we have low, medium, and high, about 50 megabyte images, but in the future, we might have super high, several dozen gigabyte images. Should the images still be part of the object or linked to, say, AWS? Also, say you have an image mask, where do you store it?
Yeah, as asked before, we just store the paths now, or the links, whatever you want, and it's up to the user to load the images. But of course, if someone has some hints to do that, to store the images into the object, we can work together to do it, and that's it.
And I think, sorry, that was kind of a long question, but the initial part of the question was how to deal with different resolutions of histology images.
Oh, I don't think it's a problem to store this kind of different quality version of histology images. If we are talking just the morphology of the cells, we can store them as data into the spatial coordinates, because any kind of object can be stored there.
All right, the next question is for Ellis. Is it possible to calculate the underlying L cross using non-Euclidean distances? I can imagine various boundary situations or curvature / manifold where Euclidean would be problematic.
I'm sure it's possible, right? So, but I suppose probably the better way to answer it is that maybe you might want to think about using a different distance metric. But one of the great things about point process models is they're a really well-established technique. And so in calculating a lot of these K cross, L cross, G cross, all these different types of summary statistics, most of them have got options at least, or people have thought about these kind of boundary situations. And so you can put weights in for edge effects. Like I said, with tissue inhomogeneity, you can put weights in terms of cell density and all those kinds of things. So I think a lot of that weighting structure that's inherent in these functions probably accounts for those kinds of issues. I don't know if that answers.
If not, I'm sure the person that submitted the question can follow up with you. So then next question for Lukas is, what clustering algorithms do you use for unsupervised analysis?
Yes, so we used the the Bioconductor Oscar pipeline, we used the shared nearest neighbor graph and walk trap clustering algorithm in there. So basically the standard single cell Bioconductor clustering pipeline. We tested out some of the parameters, like the number of clusters. We've definitely tried that out.
All right, Dario, do you see any benefit on having Alt-X for the spot level data and say pseudo-bulked layer level data, or would you keep them as separate objects?
I think that you can, can you repeat the question? Sorry.
Sorry, reading R functions isn't the easiest thing. So
Yeah.
ALT capital EXP.
Yeah, the Alt-X. Yeah.
So do you see any benefit of having Alt-X for the spot level data and say pseudo bulk layered level data?
Oh, I didn't thought about it at all. So I think a little bit about this, but I think that at the moment, maybe you can store in the alt text as it is. But of course, I have to think better about this one. Thank you.
The next question is for Ellis. It's a follow-up from the first question. So if you take the centroid, how can you account for long, skinny cells like neurons versus small, round cells like leukocytes or star-shaped astrocytes, et cetera? And there's a comment that the questioner would love to chat more and has also looked at some of these data sets. So maybe it's a good opportunity.
If there's one thing I like doing, it's chatting. I love hearing myself talk. I suppose the reality is you can't. So these things are just a point in time, but I guess it really comes down to the type of question you're trying to ask. And so if you're really just interested in like a cell, and if it's Rust neighborhood of cells averaged over all of those cells in all of the image, then I don't think it really matters too much. I think that kind of resolution where you want to see if like processes are touching different cell types, I don't think it's necessarily going to help you at these kind of big, broad, summarized association across a whole tissue.
All right, so maybe the last question I'll pose to Lukas is, what defines a layer in space? Or how would you think about it when cell types are shared across the tissue?
Yeah, so yeah, I mean, this goes to the question of what exactly is a cell type in this context as well. So if we can assume that a cell type is a layer, then that's one way of looking at it. But I think that's very difficult to answer yet. If we're interested in conditioning on cell types, for example, and then doing additional downstream analysis on that, that's a different way of looking at it. Or if we just assume that cell types are layers, yeah, I don't have a great answer to that.
All right, well, thank you to all 4 speakers again for a very interesting session. We're halfway through the contributed talk sessions for this conference, so hopefully everybody comes back tomorrow for session number 4. And thank you all.